Stability of proteins and nucleic acids. Proteins and nucleic acids are essential biomolecules in living systems that play critical roles in many biological processes. The stability of these molecules is crucial for their proper functioning of the cell. Here is a detailed explanation of stability of proteins and nucleic acids. Stability of proteins. Protein stability refers to the ability of a protein to maintain its nature, conformation, and activity under various environmental conditions. Proteins can be stabilized by several factors, including hydrogen bonds, electrostatic interactions, van der Waals forces, and hydrophobic interactions. Disrupting any of these forces can lead to protein denaturation, resulting in loss of function. Factors that can affect protein stability include changes in temperature, pH, salt concentration, and the presence of chemical denaturants or proteases. Proteins can also be destabilized by mutations or modifications that disrupt the normal folding of a protein. Chaperone proteins and proteases play a critical role in maintaining protein stability by helping to fold newly synthesized proteins or by removing misfolded or damaged proteins. Next, stability of nucleic acids. Nucleic acid stability refers to the ability of DNA and RNA to maintain their native structure and function under various environmental conditions. The stability of nucleic acids is determined by several factors, including base stacking interactions, hydrogen bonding, electrostatic interactions, and hydrophobic interactions. Factors that can affect nucleic acid stability include changes in temperature, pH, salt concentration, and the presence of chemical or physical agents that can disrupt the hydrogen bonding or base stacking interactions. So, nucleic acids can also be destabilized by mutations or modifications that disrupt the normal base pairing or stacking interactions. Next, importance. The stability of proteins and nucleic acids is critical for their proper functioning in living systems. Protein stability is important for enzymatic activity, protein-protein interactions, and protein localization. Changes in protein stability can lead to development of diseases such as Alzheimer's, Parkinson's, and cystic fibrosis. Nucleic acid stability is critical for accurate replication and transmission of genetic information, and changes in nucleic acid stability can lead to the development of genetic diseases such as sickle cell anemia or cancer. Understanding the factors that contribute to protein and nucleic acid stability is important for developing new therapies that target these biomolecules, as well as for designing stable proteins and nucleic acids for use in biotechnology applications. Metabolism of carbohydrates, lipids, amino acids, and nucleotides and vitamins. So, mechanism, initial and final products, amount of energy produced, etc. So, metabolism refers to the biochemical process that occurs in living cells to convert food into energy and building blocks for cellular components. Here is a detailed explanation of the metabolism of carbohydrates, lipids, amino acids, nucleotides, and vitamins. Carbohydrate metabolism. Carbohydrate metabolism involves the breakdown of carbohydrates into simpler molecules such as glucose and the subsequent generation of energy through cellular respiration. The initial products of carbohydrate metabolism are glucose, fructose, and galactose. Glucose is then converted into pyruvate through the process of glycolysis. Pyruvate is further converted into acetyl-CoA, which enters the citric acid cycle and generates ATP, the energy currency of the cell. The final products of carbohydrate metabolism are carbon dioxide and water. Carbohydrate metabolism produces a total of 38 to 36 ATP molecules per glucose molecule. Then lipid metabolism. Lipid metabolism involves the breakdown of fats and oils into simpler molecules such as glycerol and fatty acids. The initial products of lipid metabolism are triacylglycerols, which are broken down by lipases into glycerol and fatty acids. Fatty acids are then converted into acetyl-CoA through the process of beta oxidation. Acetyl-CoA enters the citric acid cycle and generates ATP. The final products of lipid metabolism are carbon dioxide and water. Lipid metabolism produces a total of 129 ATP molecules per palmitic acid molecule. Then amino acid metabolism. Amino acid metabolism involves the breakdown of proteins into simpler molecules such as amino acids and the subsequent generation of energy through cellular respiration. The initial products of amino acid metabolism are amino acids, which can be converted into pyruvate, acetyl-CoA, or other intermediates of the citric acid cycle. These intermediates generate ATP through oxidative phosphorylation. The final products of amino acid metabolism are carbon dioxide, water, and urea. Amino acid metabolism produces a total of 36 to 38 ATP molecules per glucose molecule. Then nucleotide metabolism. Nucleotide metabolism involves the breakdown of nucleotides into simpler molecules such as nitrogenous bases, sugars, and phosphates. The initial products of nucleotide metabolism are nucleotides, which can be broken down into their constituent parts through various enzymatic reactions. The final products of nucleotide metabolism depend on the specific nucleotide being metabolized. For example, the breakdown of adenosine triphosphate produces adenosine diphosphate and inorganic phosphate, which can be used to generate energy through oxidative phosphorylation. Then vitamin metabolism. Vitamin metabolism involves the absorption, transport, and utilization of vitamins by the body. Vitamins are essential for micronutrients that play important roles in many biochemical processes, including energy production, DNA synthesis, and immune function. The initial and final products of vitamin metabolism depend on the specific vitamin being metabolized. For example, vitamin C is involved in the synthesis of collagen, while vitamin D is important for the absorption of calcium and bone health. Then the metabolism of carbohydrates, lipids, amino acids, nucleotides, and vitamins is essential for providing energy and building blocks for cellular components. These processes are tightly regulated to maintain cellular homeostasis and ensure proper functioning of the body. Understanding the mechanisms and products of this metabolic pathways is important for developing treatments for metabolic disorders such as diabetes, obesity, and inherited metabolic diseases. Next, cellular organization. Cellular organization refers to the hierarchical arrangement of cells and their components, from the smallest building blocks to the complex structures and functions of multicellular organisms. At the lowest level of organization, cells are made up of biomolecules and organelles that work together to perform essential functions such as energy production, protein synthesis, and cell division. Those cells can then come together to form tissues, organs, and organ systems that carry out specialized functions within the body. Here are some of the important aspects of cellular organization. Cell structure. Cells are composed of various structures including the cell membrane, cytoplasm, and organelles such as the nucleus, mitochondria, and endoplasmic reticulum. These structures have specific functions that allow cells to carry out their roles in the body. Then tissues. Groups of cells that are similar in structure and function form tissues. There are 4 main types of tissues in the human body: epithelial, connector, muscular, and nervous tissues. Epithelial tissue covers surfaces and linear body cavities, lines body cavities. Connective tissue supports and connects different structures in the body. Muscle tissue allows for movement, and network tissue enables communication between cells. Then organs. Organs are composed of different types of tissues that work together to carry out specific functions. Examples of organs include the heart, lungs, liver, and brain. Then organ systems. Organ systems are groups of organs that work together to carry out more complex functions in the body. Examples of organ systems include the digestive system, respiratory system, and nervous system. Homeostasis. Cells, tissues, organs, and organ systems work together to maintain a stable internal environment in the body known as homeostasis. This involves balancing various factors such as temperature, pH, and nutrient levels to ensure that cells can function properly. Overall, estimating cellular organization is essential for understanding the structure and function of living organisms as well as the causes and treatments of diseases. Then, membrane structure and function. Cell membranes are complex structures that separate the inside of a cell from its surrounding environment. They are composed of a lipid bilayer, which is made up of 2 layers of phospholipids that are arranged tail to tail, with the hydrophilic head groups facing outward and the hydrophobic tail groups facing inward. The lipid bilayer also contains other lipids such as cholesterol and glycolipids that contribute to its structure and function. Here are some important aspects of membrane structure and function. Selective permeability. One of the key functions of cell membrane is to selectively regulate the movement of substances in and out of the cell. This is achieved through the presence of membrane proteins and channels that allow certain molecules to cross the membrane while preventing others from doing so. Then membrane proteins. Cell membranes contain various types of proteins that are embedded in the lipid bilayer. These proteins have diverse functions such as transporting molecules across the membrane, acting as receptors for signaling molecules, and providing signal transport. Then the next function include the fluidity. The lipid bilayer is a fluid structure, which means that the phospholipid molecules can move and rotate within the plane of the membrane. This fluidity allows membranes to change shape and respond to changes in the environment. Then membrane asymmetry. The 2 layers of the lipid bilayer can have different compositions with different types of lipids and proteins located in each layer. This asymmetry can have functional implications such as regulating the activity of membrane proteins. Then membrane fusion. Cell membranes can undergo fusion with other membranes such as during the process of endocytosis or when cells divide. This requires the coordinated interaction of membrane proteins and lipids to enable the merging of two separate lipid bilayers. Overall, understanding the structure and function of cell membranes is critical for understanding cellular processes such as signaling, transport, and cell division, and for developing new therapies for diseases that involve membrane dysfunction. Membrane structure and function, each with detailed explanation. Membrane structure. Cell membranes consist of a lipid bilayer, which is composed of two layers of phospholipid molecules arranged tail to tail. The hydrophilic heads of the phospholipids face outward, while the hydrophobic tails face inward, creating a barrier that separates the inside of the cell from the outside environment. In addition to phospholipids, membranes also contain other lipids such as cholesterol and glycolipids, which contribute to the membrane's structure and function. Membrane proteins are also embedded in the lipid bilayer and they play critical roles in membrane function. Then membrane functions. Cell membranes are essential for life as they play a number of important roles in cell function. Here are some of the key functions of the cell membrane, like selective permeability. One of the most important functions of cell membrane is to regulate the movement of molecules in and out of the cell. This is achieved through the presence of membrane proteins and channels that allow certain molecules to cross the membrane while preventing others from doing so. For example, channels called aquaporin cells also allow water to cross the membrane, while ion channels allow the passage of specific ions. Cell signaling. Membrane proteins play a critical role in cell signaling, which allows cells to communicate with each other. Receptor proteins on the surface of the cell bind to specific signaling molecules such as hormones and initiate a signaling cascade that can lead to changes in cell behavior. Molecules such as hormones and initiate membrane function. Cell membranes are essential for life as they play a number of important roles in cell function. They are some of the key functions of the cell membrane. Selective permeability. One of the most important functions of cell membranes is to regulate the movement of molecules in and out of the cell. This is achieved through the presence of membrane proteins and channels that allow certain molecules to cross the membrane while preventing others from doing so. For example, channels called aquaporins allow water to cross the membrane, while ion channels allow the passage of specific ions. Then cell signaling. Membrane proteins play a critical role in cell signaling, which allows cells to communicate with each other. Receptor proteins on the surface of the cell bind to specific signaling molecules such as hormones and initiate a signaling cascade that can lead to changes in cell behavior. Structural support. Membranes provide structural support to cells as they help to maintain the shape and integrity of the cell. Membrane proteins can also play a role in maintaining cell shape and function. Then energy production. Membranes are involved in energy production through the process of oxidative phosphorylation. This process occurs in the mitochondria, which are membrane-bound organelles, and involves the transport of electrons across the mitochondrial membrane to produce ATP and the enzyme currency of the cell. Then membrane fusion. Cell membranes can undergo fusion with other membranes, such as during the process of endocytosis or when cells divide. This requires the coordinated interaction of membrane proteins and lipids to enable the merging of two separate lipid bilayers. Overall, understanding the structure and function of cell membrane is critical for understanding cellular processes such as signaling, transport, and energy transduction. Production. Dysfunctional membranes can lead to a range of diseases such as cystic fibrosis and muscular dystrophy, highlighting the importance of studying membrane biology. Then structure of modal membrane. Lipid bilayer and membrane protein diffusion osmosis, ion channels, active transport, membrane pumps, mechanism of sorting and regulation of intercellular transport, and all those topics. Structure of modal membrane. The modal membrane is artificial membrane used to study the properties of biological membranes. It is composed of a lipid bilayer made up of phospholipids, which are arranged in a way that their hydrophobic tails face each other while the hydrophilic heads are exposed to the aqueous environment. Lipid bilayer is a fundamental component of all biological membranes. It is composed of two layers of lipids, which include phospholipids, glycolipids, and cholesterol. The lipid bilayer provides a barrier that separates the interior of the cell from the exterior environment. It also plays a vital role in maintaining the integrity and function of the cell membrane. Then membrane protein. Membrane proteins are proteins that are embedded within or attached to the lipid bilayer of a cell membrane. They perform a variety of functions including transport of molecules across the membrane, cell signaling, and enzymatic activity. Membrane proteins are classified into two types, integral proteins and peripheral proteins. Then diffusion. Diffusion is the process by which molecules move from one area of high concentration to an area of low concentration. In the context of the cell membrane, diffusion occurs when molecules such as gases, water, and small lipophilic molecules move across the lipid bilayer in response to concentration gradients. Next topic, osmosis. Osmosis is the diffusion of water molecules across a selectively permeable membrane from the region of lower solute concentration to a region of higher solute concentration. It is an essential process for maintaining water balance in cells and tissues. Ion channels are proteins that span the lipid bilayer of the cell membrane and allow the passage of ions such as sodium, potassium, and calcium into and out of the cell. Ion channels play an essential role in regulating the electrical properties of the cell membrane and are involved in many physiological processes including muscle contraction and nerve impulses. Then active transport. Active transport is the movement of molecules across a cell membrane against their concentration gradient, requiring the input of energy in the form of ATP. Examples of active transport include sodium potassium pump, which is involved in maintaining the concentration gradient of sodium and potassium ions across the cell membrane. Then membrane pumps. Membrane pumps are specialized proteins that use energy to transport molecules across the cell membrane. Examples include the sodium potassium pump, which uses ATP to transport sodium and potassium ions across the membrane against their concentration gradient. Then mechanism of sorting and regulation of intracellular transport. The sorting and regulation of intracellular transport are critical processes for maintaining the organization and function of cells. This process involves the selective targeting and transport of molecules to specific locations within the cell, including the nucleus, mitochondria, and lysosomes. Then electrical properties of membranes. The electrical properties of cell membranes are essential for the functioning of excitable cells such as neurons and muscle cells. These properties are determined by the presence of ion channels and membrane pumps, which regulate the flow of ions across the membrane and create a voltage difference or membrane potential. The membrane potential plays a critical role in processes such as nerve impulse and muscle contraction. Importance. Understanding the structure and function of cell membranes is essential for understanding many physiological processes, including cell signaling, nutrient uptake, and waste removal. The properties of cell membranes also play a critical role in the development of new drugs and therapies for a variety of diseases, including cancer and neurodegenerative disorders. Structural organization and function of intracellular organelles. Then, cell wall. The cell wall is a rigid layer present in plant cells, bacteria, and fungi that provides structural support and protection. It is made up of cellulose, hemicellulose, pectin, and other proteins. The cell wall also regulates the flow of substances in and out of the cell. Then nucleus. Nucleus is the control center of the cell and contains the genetic material of cells in the form of DNA. It is enclosed by a double membrane called nuclear envelope and contains nucleolus. The nucleus is responsible for DNA replication, transcription, and regulation of gene expression. Then mitochondria. Mitochondria are the powerhouse of the cell and are responsible for cellular respiration. They are enclosed by a double membrane and have their own DNA. Mitochondria generate ATP, which is used as energy source for many cellular processes. Then Golgi bodies are responsible for processing and packaging proteins and lipids for transport to other parts of the cell or for secretion outside the cell. They are made up of flattened membrane-bound spaces called cisternae. Lysosomes are membrane-bound organelles that contain digestive enzymes. They are responsible for breaking down waste materials, foreign substances, and damaged organelles within the cell. Then endoplasmic reticulum. The endoplasmic reticulum is a network of interconnected membrane-bound tubes and sacs that are involved in protein and lipid synthesis. The rough ER is studded with ribosomes and is responsible for protein synthesis, while the smooth ER is involved in lipid synthesis and detoxification of harmful substances. Then peroxisomes. Peroxisomes are membrane-bound organelles that contain enzymes involved in the breakdown of fatty acids and the detoxification of harmful substances. They also play a role in the production and breakdown of hydrogen peroxide. Then plastids. Plastids are found only in plant cells and are responsible for various functions such as photosynthesis, storage, and pigmentation. Chloroplasts are a type of plastid that contain chlorophyll and are responsible for photosynthesis. Then vacuole. Vacuoles are membrane-bound organelles that are responsible for storage of water, ions, and nutrients. They also play a role in maintaining turgor pressure in plant cells. Then cytoskeleton. The cytoskeleton is a network of protein fibers that gives the cells its shape, provides mechanical support, and plays a role in cell motility. The three main types of cytoskeleton fibers are microfilaments, intermediate filaments, and microtubules. Microfilaments are involved in cell movement and interaction, contraction. Intermediate filaments provide mechanical strength, and microtubules are involved in cell division and intracellular transport. The structural organization and function of intracellular organelles are crucial for proper functioning of the cell. Each organelle has a specific role in maintaining cellular homeostasis and carrying out various cellular processes. Understanding the structure and function of these organelles is essential for understanding cellular process and their regulation. Then the cell wall is the outermost layer that surrounds the plasma membrane of the cell. It is a rigid protective layer that provides structural support and maintains the shape of the cell. In plants, the cell wall is composed of cellulose, hemicellulose, and pectin. Nucleus is a membrane-bound organelle that contains the genetic material of the cell. It is the control center of the cell and is responsible for regulating gene expression and cell division. The nucleus is composed of a nuclear envelope, nucleoplasm, chromatin, and nucleolus. Then mitochondria. Mitochondria are the powerhouse of the cell. They are responsible for generating ATP through cellular respiration. Mitochondria have their own DNA and are capable of reproducing independently of the cell. They are also play a major role in apoptosis or programmed cell death. Then Golgi bodies. The Golgi apparatus is a membrane-bound organelle that is responsible for sorting, modifying, and packaging proteins and lipids for transport to their final destination. The Golgi apparatus consists of flattened sacs of membrane-bound sacs called cisternae. Then lysosomes. Lysosomes are membrane-bound organelles that contain hydraulic enzymes. They are responsible for the degradation of cellular waste and the recycling of cellular components. Lysosomes play a role in cellular homeostasis and the immune response. Then endoplasmic reticulum. The endoplasmic reticulum is a network of membrane-bound tubules and sacs that are responsible for protein and lipid synthesis as well as calcium storage and release. The rough ER is studded with ribosomes and is responsible for the synthesis of membrane-bound and secreted proteins, while the smooth ER is involved in lipid synthesis and detoxification. Then peroxisomes. Peroxisomes are membrane-bound organelles that are responsible for the breakdown of fatty acids and the detoxification of harmful substances such as hydrogen peroxide. They contain enzymes that catalyze reactions involved in lipid metabolism and are involved in the biosynthesis of some types of lipids. Then plastids. Plastids are a family of membrane-bound organelles found in plants and algae. They are involved in a wide range of metabolic processes including photosynthesis, the synthesis and storage of starch, the synthesis of pigments, and the metabolism of fatty acids. Then vacuoles. Vacuoles are membrane-bound organelles that are involved in the storage of water, ions, and nutrients. They also play a role in maintaining target pressure in plant cells. Then chloroplast. Chloroplasts are organelles found in plants and algae that are responsible for photosynthesis. They contain pigments such as chlorophyll and capture cell light energy and uses it to synthesize organic molecules such as glucose. Then cytoskeleton. The cytoskeleton is a network of protein fibers that provide structural support and help maintain the shape of the cell. It is also involved in cell motility, intracellular transport, and cell division. The cytoskeleton is composed of three main types of fibers: microfilaments, intermediate filaments, and microtubules. Cytoskeleton. The cytoskeleton is an intricate network of protein filaments that is responsible for maintaining the shape of cells, supporting cellular movements, and facilitating the transport of organelles and within cells. It consists of three types of protein filaments: microfilaments, intermediate filaments, and microtubules. Each filament type is composed of a different protein and has distinct properties and functions. Like microfilaments, also known as actin filaments, are the thinnest filaments in the cytoskeleton with a diameter of 7 to 9 nanometers. They are composed of the protein actin and are involved in cell motility, cell division, and maintenance of cell shape. Actin filaments are highly dynamic structures that can undergo rapid polymerization and depolymerization, allowing cells to rapidly change their shape and move. An example of a role of microfilaments in cell motility is the formation of lamellipodia, which is thin sheet-like protrusions that extend from the leading edge of a migrating cell. Lamellipodia are composed of a dense network of actin filaments that push the cell membrane forward and allow the cell to move. Intermediate filaments are the most stable and durable filaments in the cytoskeleton with a diameter of 10 nanometers. They are composed of several different proteins depending on the cell type and are responsible for maintaining the structural integrity of cells. Intermediate filaments are found in cells that are subjected to mechanical stress such as skin cells, muscle cells, and nerve cells. An example of a role of intermediate filaments in maintaining cell structure is in the formation of desmosomes, structures. Specialized structures anchor adjacent cells together. Desmosomes are composed of intermediate filaments and are anchored to the cell's membrane, providing a strong structural support that resists mechanical stress. Microtubules are the largest filaments in the cytoskeleton with a diameter of 25 nanometer. They are composed of the protein tubulin and are involved in cell division, intracellular transport, and maintenance of cell shape. Microtubules are highly dynamic structures that can rapidly polymerize and depolymerize, allowing cells to quickly change their shape and move. An example of role of microtubules in cell motility is the formation of cilia and flagella, which are long hair-like structures that protrude from the surface of cells and are involved in cell membrane movement. Cilia and flagella are composed of microtubules that are arranged in a specific pattern, allowing them to bend and generate a wave-like motion that propels the cell forward. In summary, the cytoskeleton plays a critical role in cell motility by providing the structural support necessary for cells to change shape and move. Macrofilaments, intermediate filaments, and microtubules each have distinct properties and functions that contribute to the overall function of the cytoskeleton. Examples of the roles of each filament type in cell motility include the formation of lamellipodia by actin filaments, desmosomes by intermediate filaments, and cilia and flagella by microtubules. Organization of genes and chromosomes, operon, unique and repetitive DNA, and all these topics. Genes and chromosomes are the fundamental units of heredity in all living organisms. The organization of genes and chromosomes is crucial for proper functioning of the cell and for transmission of genetic information from one generation to the next. Operon is a group of genes that are transcribed together and are involved in the same metabolic pathway or biological function. The operon is regulated by a single promoter and operator region. An example of an operon is a lac operon in E. coli, which are involved in the metabolism of lactose. Unique and repetitive DNA. Unique DNA is found in only one or a few copies in the genome, while repetitive DNA is found in many copies. Repetitive DNA can be further classified into satellite DNA, mini-satellite DNA, and microsatellite DNA. Satellite DNA is found in centromeres and telomeres of chromosomes, while mini and microsatellite DNA are found in non-coding regions of the genome. Interrupted genes. Interrupted genes have one or more non-coding regions called introns, which are interspaced between the coding regions called exons. The introns are removed from the pre-mRNA during splicing to generate mature mRNA. Examples of interrupted genes include that code for eukaryotic proteins. Gene families. Gene families are groups of genes that share sequence similarity and are derived from common ancestral gene. Examples of gene families include the globin gene family, which codes for hemoglobin proteins, and the histone gene family, which codes for histone proteins that are important for DNA packaging.